Hello, today I'm talking about Enzymes Lecture. So what we're going to learn is that enzymes help chemical reactions happen faster. And this is very important in living organisms. So let's just get right into it. Activation energy is the minimum amount of energy needed for a chemical reaction to start, similar to the energy required to push a rock over the top of a hill. So this is what we see. Here's where our reaction starts, and we have to add all this energy before we can make the reaction go. But once we get that energy, then the reaction can proceed. Okay. Once this initial energy is supplied, the reaction can proceed on its own at a specific rate. The rate of a reaction can be very slow, even after the activation energy is met, depending on factors like temperature and reactant concentration. So they're saying even after you reach this point, for the reaction to happen, it could still take a while, or it could happen very, very rapidly. It just depends on conditions. All right, let's continue. A catalyst is a substance that changes the activation energy and reaction rate. So when we say catalyst, this is a larger, more global term, where an enzyme, we'll find out, is a type of catalyst, just like a Ford is a type of car. Okay. By lowering the activation energy, a catalyst allows the reaction to occur with less initial energy and at a faster rate. And that's what you see here. Rather than needing to get all the way up here, all this energy, a catalyst allows you to only have to get this much energy. Much easier for the reaction to proceed and get started. Although they participate in the reaction, catalysts are not consumed or permanently changed, so they are not considered reactants or products. And catalysts could be used again because they're not consumed or changed. So they could do this multiple times. All right, let's continue here. Enzymes are a special class of catalysts found in living organisms. So again, we're talking about living things and enzymes being the catalyst in these living things. They work by lowering the activation energy of chemical reactions, which we just saw on the last graph, which allows reactions to happen much faster at normal body temperatures. So these are happening in our bodies at 98.6 degrees. And those are the conditions. Unlike some other catalysts, enzymes are typically proteins composed of long chains of amino acids. And I wouldn't expect you to recognize this, but an enzyme might be made up of multiple proteins all sticking together, working together like a little machine. All right. The function of an enzyme is directly tied to its unique three-dimensional structure. This is just a cartoon showing the enzyme, and then the substrate is going to be what the enzyme changes. But it's meant to give you like a visual representation. Conditions like temperature and pH are crucial for an enzyme to work correctly. For instance, high fevers can be dangerous because they can break the hydrogen bonds that maintain an enzyme shape, causing it to lose its ability to function. So you can imagine as your temperature goes up because of a fever, if it gets too high, these enzymes, actually this would be a better one, these enzymes start to unfold, they start to separate, they start to come apart, and they can't do their job. And that's one of the reasons that after a certain temperature, people die because their bodies are too hot, because the enzymes start to be in a state where they can't function anymore. In humans, enzymes generally perform best at a nearly neutral pH, which is the environment inside most cells. Okay. Now let's look at the actual functioning. The function of an enzyme is determined by its specific shape, which allows it to bind to certain molecules called substrates. So we see down here, and this can actually go in either direction, but we see down here the enzyme in blue, and then the substrate is just whatever chemical or molecule it's changing. But the enzyme, now there's thousands of different enzymes, because the enzyme is specific to this specific substrate. It only changes that chemical, not other ones. There are other enzymes that will change other chemicals. All right. This interaction is often explained by the lock and key model, where the substrate, here, the key, fits perfectly into a specific region on the enzyme called the active site, the lock. That's what we see going in here. Substrate is the only chemical that can fit in here. When the substrate binds to the active site, the enzyme can then weaken the bonds within the substrate molecules. So now it grabs onto that substrate and it's kind of twisting and turning, weakening the bond between the substrate. Eventually, the idea is that it breaks that bond, separates these two, and that was your chemical reaction. Also, like I said, it could go the opposite direction where you take two things, put them in here, and bind them together. Enzymes play a dual role in speeding up reactions. They bring the correct reactant molecules closer together, and they lower the activation energy. While the lock and key model is a useful analogy, a more modern understanding is that enzymes and substrates don't have a rigid fit. So that means they're not a perfect fit like a lock and key. It's very close, but having a non-rigid fit is going to matter here. Instead, the enzyme's active site, this area, slightly changes shape to fit the substance, a process that helps to strain and weaken the substrate's bonds, making them easier to break. That was that kind of twisting and pulling I was talking about. This bending of the enzyme is one way bonds in the substrates are weakened, requiring less energy for the reaction to occur. Okay, I hope this gets you started on enzymes. If you have any questions, let me know. Thank you very much.